Okay, so while you're getting your microscope lab file open and getting set up here, let's quickly talk about what Hooke did here when he invented the first microscope. Okay, so Hooke's first microscope was a lens series. Okay, and that is also what you have. Okay, in your light microscopes, you have your objective lens. Okay, which is the ones that are on the rotating nose piece. Okay, and then you also have the ocular lens. Right? So these two lenses work in concert, and what lenses do is because of their shape, they change the speed and thus the path of the light that's coming through them. All right, so if we simplify this, okay, to look at just one lens, okay, so if we have, let's say, a convex lens, okay, so that's one that looks like this. When light comes into that lens, okay, so let's say that here's our specimen right here. Okay, actually I'll draw something like, I'm just trying to think of a good shape I could use here that I could actually draw. I'm going to draw this little cloud looking thing, okay, and it's going to have a dot over here. All right, so it's like a cell, okay. Now, the light from the light is obviously going to go through the specimen. It's going to travel through the specimen, and it's going to travel, as all light does, in straight lines to the microscope. Okay? When it hits the lens, because the lens is made out of glass, glass transmits light at a different speed than air does. All materials do this to light, okay? They slow light down. The more they slow light down, the more the path of light is bent, all right? So glass bends it a fair amount. Diamond actually bends it almost more than anything else, okay? It's actually how a jeweler can tell the difference between a cubic zirconia and a real diamond, okay? All they have to do is look at the angle that the stone is cut at. Okay? In order for cubic zirconias to sparkle like a real diamond, they have to be cut at a different angle than a real diamond does in order to make the light disperse the way it does. Okay? So all materials refract and bend light a little bit differently, called their index of refraction. Okay? So this light that comes from the specimen hits this lens. Okay? When it hits this lens, it travels through it, but its path is altered. I'm just going to move this down so I can show it a little bit better. Okay, so the path of light as it goes through a convex lens, okay, is going to be diverged like this. So the light will go through the lens, but after it goes through the lens, and this one that goes through the middle, it won't have its path altered because it's going through the center part. Okay, so I've got these 5 lines here. They each come from a different part of the cell. Okay? When that has gone through the lens, okay, the light that came from this part of the cell now ends up way over here. So when I'm now looking at my specimen, let me give myself enough room to draw it here. Okay? What I'm going to see is this exact specimen here. I still didn't make it big enough, but that's okay. No, I didn't flip it the right way. Sorry, guys. I forgot I could do this. It's been a while. Okay, so that's has to go this way. Flip. That's okay. All right, so the light that came from this little bit down here, okay, is now over here. Everybody with me on that? I didn't put the circle in here, but the little circle would be right Okay, so I've got this thing flipped now, but now it's bigger. Okay, so what the lens has done is now it's making it look a lot bigger because it's diverged the light. Okay, it's spread out that same amount of light that went through this tiny little microscopic specimen and made it take up more space like this. Kind of follow? Okay, so a microscope works by bending the path of the light that comes through the lenses in such a way that it makes objects bigger. A telescope works the same way. Okay? It's going to take light that comes from a distant object. Okay? It's going to put it onto a mirror, a curved mirror, that's going to diverge the light so that when you look at it, it appears larger. Everybody okay with that? Okay, so that's how a microscope works. The reason I've just explained that is because you have to explain that in the lab. Okay? In your own words. Okay? Does everyone kind of follow how the bending of light, path of light changes? Okay, this is called, okay, where did I lose the time?
You said you flipped it, but it is the same.
Okay, so this little thing here, it was on the top of this before, right? And what a lens does is when it goes through two lenses, the image actually ends up getting flipped over, inverted. Okay, it's what we call a real image, so it appears like backwards. So if I was looking at a letter E, like we're going to do tomorrow, I'll put it in right side up. So I look at it, it looks like a letter E. I put it in the microscope and it's going to look the other way through the microscope because going through two lenses will cause it to flip over. All right, so path of light gets altered. That's basically all you need to know. Okay, it's called refraction. Okay, understand that lens, that's what lenses do in order to magnify an image. Okay. All right, so the problem we're investigating, how do microscopes allow us to observe or see tiny specimens? Okay, remember that you need to reword the problem in your own words in the form of a question. So something like that, except not exactly that. Okay, it would be fine for you to rewrite the problem. Now, for our design, here's how this lab is going to work. You are going to be looking at 4 different specimens. Okay, those 4 specimens, are a letter E, an amoeba, a paramecium, and a euglena. We're going to start out big and we're going to get smaller and smaller and smaller. Okay, the letter E, obviously quite large, about 3,000 micrometers. Okay, we're going to move down to an amoeba, which you can actually see on the slide with the naked eye, like a little purple dot. Okay. When you put it under the microscope and view it, it'll look quite a bit different than that. Okay. An amoeba is usually somewhere around 300 to 400 micrometers. Okay. Then you look at a paramecium, that's around 200 to 300 micrometers. And then you look at a euglena, which is somewhere around 80 to 100 micrometers. They're quite a bit smaller. Okay. So it'll get kind of more and more challenging as we go to look at, find, and focus the smaller specimens. Right? So, if we're thinking about manipulated, responding, and controlled variables, I would say that our manipulated variable probably has something to do with the fact that we're looking at different things each time. Okay? You've got to find a way to identify that and explain it. Now, for this lab, the purpose of this lab is to look at how a microscope works. We're looking at a bunch of different things, and we're hoping that their appearance is going to be somewhat different. Okay, I would say that that probably has something to do with our responding variable. Right, so find a way to identify and explain that. Now, we need 3 controlled variables. So I want you to think about what will be the same each time you look through the microscope. Okay? These can be procedural controlled variables, or they can be conditional controlled variables. Okay? I would suggest that you are probably going to use the same technique each time you look at something. Okay? You would probably need to describe that technique, okay, and why it's important that you do it the same for every single thing you look at. conditional controlled variables would be anything that has to do with the responding variable. Anything that can affect the appearance of the specimen would need to be controlled, okay? Think about some of the things that you can control with the microscope that would affect the appearance of the specimen, okay? I'm thinking of some of the things that you can adjust on there, like light, stuff like that. One thing that will not be controlled is magnification. We will be looking at all of these things under different magnifications, and that's just because they're all different sizes. Okay? It does no good to look at the letter E on the high power lens because you can't see anything but a tiny little portion of it. Okay? Similarly, it does no good to look at the Euglena on scanning power and try and make a drawing of it because it's too small. Okay? It looks like a little tiny blue sliver, okay? and you just don't see enough detail. So don't mention magnification as a control variable. It is not. Okay? It is also not a variable we're going to identify at all. Okay? Magnification is something that will be different, but it's the nature of what we're doing. Okay, for our hypothesis. The if part of your hypothesis needs to talk about how a microscope works. Luckily, we just went over that. Okay? So in the if part of your hypothesis, you should talk about what we just talked about, and the path of light and lenses and how lenses can alter that, and okay, things of that nature. So your if part would go, if a microscope works by blah, 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 about what I just did up there. Incidentally, I did record it, so you can go back if you missed it. Okay? The and part, as always, describes the experiment. Okay? Various specimens are going to be viewed through the light microscope. The then part needs to tell me what you will see. Okay? And by what you will see, I mean what is the appearance of these specimens going to be if the microscope works the way we say it's going to work. Okay? So if the lenses do their thing, all the specimens we look at should have these properties when I look at them through the microscope. So the procedure for the lab is going to work like this. Okay? When we go into the lab, you're going to go and grab a microscope, you're going to take it back to your table, and you're going to plug it in. Okay? Then one member of your group will come up and get the first slide. Well, actually, you're going to get the stuff to make the first slide because you're going to make the slide of the letter E. Okay? How many people have ever made a wet mount before? Not many, so this will be good. Okay, here's how a wet mount works. Okay, to make a wet mount, you're going to take a little glass microscope slide. Okay, so it's just a rectangular piece of glass. Okay, so it'll look like this. Right? You're going to take that rectangular piece of glass back to your table, and you're also going to grab a plastic cover slip, which is just a square piece of transparent plastic. Okay, they'll all be up at the front. Also up at the front will be scissors and newspaper. And what you will have to do with the scissors and newspaper is you will have to cut out a lowercase letter E from an article, not a headline. We want it small. Okay? So you're going to cut out a little piece of paper, okay? With a lowercase letter E. You're going to take all that back to your table. And an eyedropper with some water in it. Okay? You're going to place the letter E on the slide, and then you're going to put a drop of water on it. Now, caution. If you don't hold the E down when you do this, the E will flip over and go everywhere. Okay? It'll just try and float on the surface tension of the water. Okay, that whole polar nature of water thing we talked about, right? It's going to do this little dance. It's going to be the wrong way. You're not going to like it. Okay, so what you're best to do is hold it down with a pen or a pencil and then apply the water. That way it stays in the position you want. So you'll have a drop of water on here. The letter E will get all wet, and that's fine. Okay, and then it'll be there on the slide. Then you're going to take the cover slip and you're going to gently drop it on. And here's how you do that. Now we're looking from the side. Okay, so your letter E is like right here. Okay, you're going to take that cover slip, that square piece of transparent plastic, and you're going to set it at an angle on the slide like this, and then you're just going to let it go, and it'll drop down, and it'll squish the water to the edges. Okay, and it'll pin the letter E down. Okay, that cover slip won't come off. Why won't it come off? Think about that whole polar nature of water thing again. Okay, so the cover slip will come down and your slide will be set up. You need to put your slide in the microscope this way, with the letter E right side up. Okay. Then you're going to view it on scanning power only. Okay, you only need to look at the letter E on scanning power. Really, the only reason we're looking at the letter E is because it's a known shape. And when we look through the microscope, it's going to have a different orientation, and that can help us to confirm this whole idea about what the lenses do, okay? How the light behaves when it goes through. All right, that's the only reason we're looking at the letter E. Once you have the letter E focused on scanning power, you need to take a picture of it through the microscope. Okay, so you're going to take your phone, and you're just going to put it up to the lens. Okay, it'll take a little bit of fiddling. Okay, you got to get it right, get it so that you can see the letter E clearly. You can, magnify and pinch and whatever you need to do to get it focused. Okay, tap and hold, okay, get it focused, get it good, okay, and snap a picture of it. You're going to be using those pictures for your lab diagrams. Okay, we're in the digital age. You don't have to do these by hand like I did. Okay, because pre-digital cameras, we actually had to draw by hand, and I suck at drawing really bad. Okay, so my lab diagrams are very bad. Okay, you guys get to do lab diagrams with actual pictures, so it's much better. All right, so you'll take that picture and then you'll use it to make your lab diagram, which I'm going to show you how to do in a little bit. All right, so that's your first one, making a wet mount of the letter E. All the other slides are already prepared. You just have to grab them and put them under the microscope. Okay, when you've got that picture taken, then you'll put the cover slip and the letter E in the garbage, and you'll rinse off and dry the glass slide and put it back. All right. Okay, let's look at, where are they? Okay, here are the other three things we're going to be looking at. Okay, the other three things we're going to be looking at in this order are the amoeba, which will be, depending on the stain, this color to darker purple to blue even. Okay, but they generally are a blob. Okay, kind of a shapeless blob. No, you'll be able to see obvious structures within them, but they're going to be a purplish to blue blob. Okay, you need to get that on at least the low power lens. The middle lens is probably the best lens to get the picture of the amoeba. When you go to high power, you'll have trouble focusing the amoeba, because the amoeba for a microscopic organism is actually pretty large, and it's not two-dimensional. It's got depth, and it has enough depth that on high power, you can't focus the whole thing. You focus like the closest part, and the last part is out of focus, kind of like this. If I'm looking at Jazz right now, okay, everybody behind her is out of focus. But if I focus on Marcus, Jazz is out of focus, right? Same thing happens with the microscope, okay? The amoeba is thick enough that I'll only get parts of it focused at one time, and then it's hard to get a good picture of it, so on. We want to take a picture of it on low power, okay? Instead, we'll see better detail there. Ready with me? That doesn't mean that if you want to look at it on high power, you can't go right ahead, but your best opportunity for a picture is on low power. Okay, the second thing we'll look at is the paramecium. Okay, the paramecium generally have this kind of oblong shape to them. Okay, they live in pond water just like amoebas do, but they're a little bit smaller. And if you're really lucky, you'll even see this kind of blurry thing here around the edges. That's their cilia. They look like little hairs. Okay, and it's what they use to move. Now, fortunately for us, all of the things we're looking at are long dead and preserved on the slide. Trying to see a paramecium when they're alive is very difficult because they move very fast, okay? And they just go zipping across the field of view in your microscope. So you actually have to put a drop of glycerin, which is like got the same consistency as pancake syrup, on the slide to slow them down. Otherwise they just go zipping around and you can't see them very well. These ones aren't going to go anywhere because they've been dead longer than you've been alive. Okay? Those slides are really old, so they're not going to go anywhere. You'll be able to see them. Depending on the slide you get, they can vary in color greatly. Okay? There's brown, there's pink, there's blue, there's purple, even some that are green. Okay? So you'll see all different colors, okay, of those. And again, you want to get a picture of these ones, but these ones you want to get a picture of them on high power. Okay? They're small enough that your best opportunity for a picture of them is high power. All right, the last thing you're going to look at is the Euglena. Now, this is way bigger than you are going to see it. Okay? When you look at the Euglena on the scanning power lens, it will be a small faded blue sliver. That's it. They're very, very small. Okay? When you get it on low power, it will be a slightly larger pale blue sliver. Okay? And when you get it on high power, it will look kind of like this, but more like that. Okay? They'll be quite small. Right? You're not going to see a lot of detail within the Euglena just because they are so small. And this, for a lot of you, is one of the first times you'll have ever used a light microscope, okay, to look at something that small. Okay, get a picture of it on the high power lens as well. No question? Yep. All right, everybody with me there? You good with that? Okay, so those are the four things: the letter E, the amoeba, the paramecium, and the Euglena that we'll be looking at under the microscope tomorrow. Now, what you're going to have to do with those pictures is make a lab drawing or a lab diagram. Okay, we'll talk about that in a minute here. Okay, and then you've got your analysis. So your lab diagrams are going to be your observations. Okay, then you're going to have these analysis questions. Okay, question number one, using what you saw under the microscope, size, orientation, general characteristics, explain what you think happened to enlarge the specimens. Basically, explain. Okay, what happened to the light after it passed through the specimen, went through the lenses? Why do these things look the way they do? Okay, second question, describe and explain the steps you took to find and focus your paramecium. So walk me through what you did step by step to focus the paramecium. Okay, and then the third question, why do we add the water to the wet mount? Why don't we just put the cover slip on the top and go from there? Okay, talk about why we might do that. Then your conclusion, same as always, copy and paste your hypothesis and then explain based on your observations whether you can accept or reject it. Okay, and then any sources of error and ways to eliminate them. I would accept as a source of error here, inexperience could be a source of error. Okay, just explain to me how that could affect your ability to get good shots of the things we're looking at. So I'm giving you one of the sources of error. You just have to explain it. Okay, you have to come up with one more. Okay, let's talk about how we do the lab drawings, okay, the lab diagrams here. So I'm going to sit down at my computer here and kind of walk you through the steps.